see him, and he said yes.
Right.
So just for everyone's information, we're now live streaming on YouTube and being recorded. So we got to watch what we say. Should we start now, Phil, do you think?
Sure.
Okay.
Sure. Well, since Christian's not here, it'll fall to me to introduce our speaker tonight. It's been my pleasure to cross paths with Danny on several occasions, both down here and up in Oregon. So besides having a whole lot of other credentials, Danny is one of the authors of
Mycomatch is our new name.
What's what?
Mycomatch is the new name now.
Okay.
Formerly Matchmaker.
Formerly Matchmaker, yes. Wonderful piece of work, incredibly able to do so many different things. So the last time Danny and I crossed paths, it was up in Oregon. And I asked him a simple question, is like, when could we have this thing working down in California? The answer was something like, you got all of those oaks down there. You don't know how much work it would be to put in all of those data. Oh my God. So, anyway, with that, I'll turn it over to you, Danny.
Thank you. Thanks so much for having me here. I'm really excited to come and talk about a topic that is just becoming more and more popular all the time. People are really curious what's going on with DNA sequencing. It's one of the things responsible for people having to learn a new name for all their favorite mushrooms every couple of months. So that can be frustrating. And people kind of want to know, is it ever going to end? Are we going to keep changing these names, or are things going to settle down? Well, things are going to settle down. It's just taking a little longer than we thought. But I'm here to talk today about the biggest revolution and what it really means in the larger context of mushrooming at large, and sort of give a peek into a day in the life of what the modern mycologist is doing, starting all the way back with what mycologists used to do hundreds of years ago and how this job has changed. So as human beings, we love to classify things. We went out and discovered all thousands of different kinds of mushrooms that there are in the world, but it's not enough just to have a big book that lists thousands of mushrooms that are around. We want to put them in boxes. We want to classify them and say which ones are related to. Since the dawn of time, we've made diagrams like this tree to try and guess how things might be related. So like these two species would have a common parent or a common grandmother or grandfather, and this would explain how they're all related to each other. So it all started back in the 1700s. And in the beginning, before we had a lot of technology, the only thing you could do is you could look at a mushroom and you could say, that one looks like this other mushroom. Maybe they're related. And this one looks nothing like this other. That was really the best you could do. So Elias Fries, who wrote some really groundbreaking books starting in 1821, was the first guy to really make his life effort to classifying how these organisms were related to each other. People had been describing mushrooms before, and we had names for these things, but nobody really tried to figure out how they all related to each other until he came around. And he decided to group them by their most obvious property. It was a regular gilled mushroom. You look under the cap and you see the gills. He said that we'll call agaricus. Those are the gilled mushrooms. And then some mushrooms like the porcinis, you look underneath the cap and you see that they have the sponge or the pore surface. He thought that was very, very different kind of mushroom. He called those boletus. So that was the beginning of classifying mushrooms into their various groups. And for the guild mushrooms, he decided the most important thing to know to tell the guild mushrooms apart was what color are the spores. So at 8 hours to take a spore print before an identifier helps you identify what your mushroom is, this is the guy you can blame. Fries is the guy who decided that spore color was going to be the important thing that everything would be labeled under, and you have to know it in order to be able to get anywhere in the books. So he's the guy to blame for all the time you have to spend taking spore prints. Now, honestly, it is a very informative property. It's true, but it certainly can be a hassle sometimes to deal with it. So when they were classifying things, they sometimes had to make some tough decisions. On the left here, you have obviously an agaricus. It's a regular mushroom that has gills under it. Also, the guild mushrooms are usually soft enough to eat. They're not very chewy. They won't break your teeth to bite into them, and they often grow on the ground. Another classification of mushrooms on the far right, the polypores or the conchs. Those are hard and tough. They grow on wood, and if you tried to bite into one, you would break your teeth. So we had two really good different categories here. And then along comes the mushroom in the middle. This gloeophyllum. It's a hard, wooden, tough conch that grows on the side of trees. But as soon as you turn it over and look underneath, you see it has gills underneath, which is really surprising. So it has some properties of both. So what do? Is it an agaric, a guild mushroom that just happens to be tough and wooden? Or is it a conch that just happened to evolve gills underneath instead of pores? Which way do you go? Well, Fries's rules that he had set up said that the most important classification was what's underneath the cap. If it's gills, it's an agaricus. If it's hard wooden conch and there's pores underneath, it's a polyporus. So it was classified in agaricus. But it's just kind of a matter of opinion. Was he right? Was that the right thing to do? Nobody really knew for sure, but you just had to make a decision somehow, and that's what they did. So the second big revolution to come along was the invention of the microscope. And as soon as you started looking at these mushrooms under the scope, they found some really surprising, fascinating things. When you look at most guild mushrooms, you see that their spores are ellipses. They're not perfectly round. They're longer than they are wide, and usually pretty smooth. And all of the other tissue that you see besides the spores, you saw a lot of long, stringy things, like in the middle picture, if you can sort of see in the background behind the spores. It's just a whole bunch of long, stringy, fibrous things. And that was pretty much what you saw on every guild mushroom until somebody took a russula and put it under the microscope and was really surprised. They're like the shrimp russula. The short-stemmed russula is something that turns into a lobster mushroom if it gets infected by the right parasite. So these russulas under the microscope were fascinatingly different. Not only were the spores perfectly round, as you can see, but if you put iodine on them, all of these warts that are on the spores turn black. It's covered with black bumps, basically. And when you look behind the spores at all of the other tissue that's in the mushroom, it wasn't just all long, stringy things. There were a whole bunch of other circular, elliptical shaped things too. Lots of circle shapes everywhere, and these bumps in the spores that turn black. So very, very different than any other guild mushroom. And they started thinking, something is up. This is not at all like what any other mushroom was. And they realized something very important, which was differences that are invisibly small might actually be much more important, and much more differences that are obvious to the naked eye. Just what you see with your own eyes can be less important. So for instance, whenever they looked at porcinis and boletes under the microscope, they had very long, narrow spores, usually 3 times longer than they were wide, like nowhere near spherical. And then when they looked at what we call the guild bolete, Phylloporus, under the microscope, they saw the same thing. Now, this was already a weird mushroom because you come at it from a distance and you think you found a bolete. You see this bright yellow underneath. You see this kind of a felty top, and just the whole sort of gestalt of it looks like exactly like so many different boletes. And you turn it upside down and there's gills where there's supposed to be pores. So people wondered if that was a coincidence that it looked like a bolete, or if it really was some kind of weird bolete with gills. Well, Fries's classification said that it had gills, therefore it was a gilled mushroom, and that just happened to look like a bolete. But when the microscope was invented, they realized they got that backwards, because everything under the microscope looked exactly like a bolete, and that answered the question that this was actually related to the boletes that had just happened to evolve. Those gill mushrooms that look to the naked eye like every other gill mushroom in the world, they're a third completely kind of category of mushroom altogether, because they were so different. So they started finding some of these truths here. And so they went and they classified every mushroom. They ended up relaxing Fries's rules. So if the microscope showed that similar looking mushrooms really were very different, or different looking mushrooms looked exactly the same under the scope, they moved them around and they categorized them in a way that they felt was more real and logical and got closer at the truth. And they abandoned the arbitrary rule of, if it has gills, it must be related to other gill mushrooms. But still, did they get it right? And is there really even anything as a right answer? Maybe this is all just a matter of opinion. You can see something that looks like it's in between two mushrooms. One person thinks it's more like the first one. Another person thinks it's more like the second one. Perfectly valid opinions to have. So people started to wonder if there even was a right answer, or if they should just keep arguing about it forever. And then finally, we discovered DNA and we got to the real answer. So it has basically been officially decided that if you are genetically related to something, you're an actual relative of something, that means that you are related, and that is how we should classify things. So in other words, if two mushrooms have the same great grandparent, then they are closely related, and that is how the tree of life that we build that shows relationships should actually reflect that. And everybody's pretty much accepted this because it's a very useful way to do it too. Who cares if somebody thinks that two mushrooms look alike and they want to say that they're related? That doesn't really have any meaning. But think about this. If you have a very dangerously poisonous mushroom and you find something that is genetically related to it very closely, it might also be dangerously poisonous, and you should look out for that. If you find one mushroom that has lots of these medicinal properties and health benefits, and you're looking for other mushrooms that might have those same properties, relatives, to see if they might have the same properties. This is a very useful way to thinking about it. And this is the way that it has been decided it's really going to be done. So let's talk a little bit about what DNA really is. DNA is a very long string of molecules that you can represent by a very long string of letters. So inside your cells are 4 different types of molecules: thymine, cytosine, horribly mispronounced, and we've color-coded them, and we just call them by their first letter, T, C, A, and G. So on a DNA strand, this double helix here that you may have seen before, you'll have color-coded showing you which molecule exists there. And as you read in the direction of the arrow, going from the bottom here, spinning around to the top to follow the arrow, you can just look at the color of the molecule that's attached to that strand and read off the letters, and that's your DNA sequence. So you might read A, C, G, T, C, C, blah, blah, blah. Describes everything there is to know almost about an organism. So they're 50 million characters long for a mushroom. It takes about 50 million characters to describe a mushroom. Well, humans are much more complicated than mushrooms, as you may have guessed. And there are about 3 billion characters of DNA in a human, which is a very, very long string. And we actually have 23 chromosomes, which means we don't just have one long strand of molecules, 3 billion long. We have 23 different strands. And the total length of all 23 of those is about 3 billion long. So it's really interesting that a DNA sequence has an analogy like it's a computer file or a computer software program. And if you think about DNA as a computer file, it actually works really well, and it's kind of scary. For instance, in a computer file, if you have a thumb drive and you have some pictures on it, some GIF pictures on it, and you look at that actual file, the first 3 letters in the file are GIF, which is what tells the computer, this is a GIF file. Look for a picture here and put it on the screen. So it will. If you have a spreadsheet, an Excel spreadsheet, and if you take a look at that file on your thumb drive of your spreadsheet, every single spreadsheet file starts with the characters D0CF11E0 blah, blah, blah. And that's the way that it announces to the world, I'm a spreadsheet. So think of me as a spreadsheet. Don't try to draw me as a picture. Look at me as a spreadsheet and get the data out of it. So it's kind of the same way with DNA. When you're at the part of the DNA that is going to say what your eye color is, you will always find the string, for instance, AGCTTAG, blah, 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 TCA. And when you see that string in your DNA, that has meaning that says the next thing is going to indicate your eye color. So it's kind of really like a computer file. Now, when humans count, we use base 10 because we have 10 figures, so that was pretty handy. We have 10 different digits from 0 through 9 when we count. Computers are binary, which you may have heard. They're only base 2. 0 and 1 are the only numbers that computers count with. They're just 0, 1, 1, 0, 0, 1, and those are the only numbers. So biological organisms use base 4. They have 4 digits, A, C, G, and T, 4 different digits that they can have to represent what they are. So that's kind of interesting. Now, software without hardware does no good at all. If you have a thumb drive with a picture on it, you can't take a look at that picture. You can't look at that thumb drive and see what that picture is. You need a computer which is a machine, to look at that file and actually do the work of interpreting that picture and putting it up on the screen. Well, the analogy holds. Your body is the hardware, or is the computer that reads the software in your DNA to figure out what to do, what color to make your eyes, how many fingers you're going to have, all of that kind of stuff. So the sort of computer analogy to DNA is a very interesting one. So one kind of interesting fact is that you don't just have one set of DNA inside you describing who you are. There are 2 complete sets of DNA inside each person, which means there's basically a description for 2 different people inside you. And it's mostly the same. The sort of description of the 2 people inside you have very small differences. For instance, in this chart here, I put an X to show where the differences are. One has an A and one has a T on top. And then about in the middle here, you can see one has a C and one has an A. But besides that, they're the same. So they're mostly the same, but there are some key differences. So let's say person number 1's DNA description inside you is that of a brown-haired person, and person number 2 is that of a red-haired person. Well, which are you going to have, brown hair or red hair? What is the real unique combination of the 2? And you've probably heard about dominant genes and recessive genes. Well, brown hair is dominant. So if one of the descriptions inside you is for brown hair and one's for red hair, brown hair kind of wins out and it is dominant. So you will have brown hair. And there's a fun fact that if you are a genetic male, one of the 2 people inside you is the DNA code for a genetic female. So the way sexual reproduction works, which is very interesting, is random parts of the gene are chosen from the 2 sort of people inside you to determine what you're going to pass on to your offspring. So for instance, you may have one of your strands of DNA describes brown hair and the other one describes red hair. It will arbitrarily say, pick the brown-haired person and send those genes on. And for left or right-handed, it'll pick one at random. It might pick the right-handed one. For boy or girl, it might pick girl. For I love cilantro or I hate cilantro, it might pick I love cilantro. And it knows where there's a useful segment of DNA, and it will only switch after each sort of piece has been expressed. It's not going to switch back and forth from left to right at any arbitrary point. That could give extremely mixed up DNA that you can end up with 3 eyes on the back of your neck or something. And we can't have that happen. So it's smart enough to know which set of letters represent one trait and to copy one of the 2 copies of that trait over. So this new single strand of DNA that is created by combining the 2 that are inside you goes either into your egg or into your sperm. And when it's combined with somebody else's, you get the 2 strands back that make up an entire person. And that's how sexual reproduction works on the DNA level. So we want to sequence our mushroom or our person or anything. And how do we do that? Which part do we sequence? 50 million for a mushroom is very, very long. If we were to sequence all 50 million base pairs, that would be extremely expensive and a waste of time because it would take so long to look at. We do not need it all. So typically what we do is we pick just one tiny short piece of that 50 million that is going to tell us whatever it is we need to know. And one of the regions to pick is the ITS region, internal transcribed spacer. Now, this is a kind of what's sometimes called a junk DNA region, which means it doesn't do anything. It's actually a part of the DNA that's never looked at. It can be anything and it doesn't matter. So it's free to mutate because DNA does mutate all the time. And any mutation that happens here is going to be harmless. It doesn't have any chance of killing the organism or making it better or worse or anything. It just doesn't matter. So since it is allowed to mutate freely, it will mutate faster than other parts of the gene. And if you want to see if 2 mushrooms are the same species or very closely related or not very closely related, this is a really good piece of DNA to look at. It helps you tell if 2 organisms are the same species or not. And only mushrooms have an ITS part of their DNA. We do not. So you don't have to worry about being in a sterilely clean lab and not contaminating it. You can be taking your pieces of mushrooms and handling them with your hands, putting them in the vials, and your DNA getting in there will not contaminate it because your DNA is going to be ignored because it's only going to be looking for ITS DNA, which is a piece of DNA that's in mushrooms and not in humans. So DNA sequencing, let's talk for a minute about how it actually works, because I think this is very interesting. It has a PCR, which is a chain reaction, and what it does is it makes billions and billions of copies of just the little piece of DNA that you're interested in. So you'll end up with a vial. Instead of having all the DNA of it, you'll have billions of copies of just the little piece that you want. And it's kind of a recipe. So here's the ingredients for our recipe. We need a piece of the mushroom, check. You need a forward primer, which means you need to know what letters you'll see in the DNA that basically announce, okay, this is the beginning of the ITS region that you care about. There's a particular letters there. And when you see those letters, you know for a fact that the next thing coming in the DNA strand is the ITS region you want. So you know what the beginning of the region looks like. Then you have the reverse primer, and that is the end of the region you're interested in. It's the characters you see after the ITS region is finished. So you know what those characters are, so you can look for just the piece you want. And the next thing you need is something called the polymerase enzyme. And it's this enzyme that if it's present in your sort of soup, it will trigger DNA copying. And so you have to have the enzyme present. We'll talk about that later. And the last thing you need are a whole bunch of spare letters, spare molecules that represent A, C, G, and T, just a whole bunch of each of those individual letters floating around in your soup. So here's a picture of what it looks like. So here's your soup at the top. We see floating around are a whole bunch of individual letters. We have the enzyme, that's this funny little cartoon here. This is this enzyme that allows DNA sequencing that allows DNA duplication to happen. You have your very long strand of DNA. This is your piece of mushroom. You've put your mushroom in your vial, and it has arrows on both ends because it's a very long string, 50 million letters long, going forever in both directions. And you have which match up with the beginning and the end of the string. Okay, so step one is you heat it to 95 degrees Celsius, and at that almost boiling point, the DNA strands will separate. They'll kind of unravel like a zipper. So each double strand will break in half and become 2 single strands. Step two, you set things to 55 degrees. And at 55 degrees, the primers will find the beginning of the piece of DNA that you're interested in and attach themselves. So these 2 vertical black bars represent that it's everything in between these bars is the ITS region that we want to sequence. That's the part we care about. We care about the part between the black bars. And then to the left and to the right of the black bars, they go on for 50 more million characters that we just do not care about. So at 55 degrees, the primers have attached themselves to the beginning and the end of the part we care about. And the final step, part 3, is you set it to 72 degrees, and the enzyme will do its work and start duplicating the DNA. All of the loose letters will find the place that they fit in, and they'll snap into place one by one and go along, and all the little letters will snap into place and create 2 full strands of DNA again, and you have now copied 1 piece of DNA into 2 pieces of DNA, but you've only copied the part that you care about. So it turns out all these little letters are able to find the right place to go to about 1,000 every minute. And since the length of the ITS region is about 700 or so, it's less than 1,000 long. So all of the pieces will have found their spot, and you'll have duplicated what you need. So you keep it at 72 degrees for about 1 minute, and you're done. And then we keep doing that over and over and over again. We go back to step 1, set it to 95 degrees, and they'll split and part again. Set it back to 55 degrees. The primers will find their spot that they're supposed to go in, and then set it to 72 degrees again, and it'll duplicate those. And every time we go along, it will copy 2, 4, 8, 16, 32. And by the time we've cycled 30 times, which can take anywhere from hours, you will have more than 1 billion copies of the DNA set. And almost every single strand that you'll have will be just the piece that you were interested in. So let's talk a bit about this magic enzyme that does its work. So one problem with early DNA sequencing is that DNA replication, your body is duplicating your DNA constantly as part of being alive. So it happens at room temperature. So it's very important that you do not have any DNA being duplicated until step 3, right? And you don't want it to happen in step 1 and step 2. So all of the enzymes that we know of and all of the people and animals and plants that live at room temperature, they're constantly reproducing the DNA. And the only way to stop them is to go into sub-zero temperatures. And you used to have to go into a frozen meat locker where the temperature was like 40 below zero and be freezing your butt off while you do these experiments, because it had to be kept super cold, because the moment it warmed up, it would all start duplicating. So what they needed to do was to find an organism who lives near the boiling point of water, and that's regular temperature for it. Well, in the geysers in Yellowstone, like Old Faithful and so, they found these bacteria that live at 72 degrees Celsius. That's their regular body temperature. And their enzymes duplicate DNA only at those really high temperatures. So we used those enzymes in the DNA process so that we could be comfortable in our lab, in our homes, in our kitchens, and do this at room temperature without having to walk into a meat freezer. So that was pretty handy. And the reason it's called the TAC polymerase is because that's the name of the bacteria. Its Latin name is Thermus aquaticus, or T period AQ period. So its nickname is TAC. So we call it TAC. So it's time to watch an animation. Hopefully our internet will be able to see this. Let me share the this part of the screen here, and let's watch this movie. So here we are, step one. We set it to 95 degrees, and the DNA splits apart like a zipper. There's our enzyme getting ready to do its work. Step two, we set it to 55 degrees. And the primers find the beginning and the end of the part we're interested in. It's just this middle part that we care about. Step three, set it to 72 degrees. And thanks to the presence of that enzyme, all the loose floating letters find their spot and snap into place. And we've duplicated the DNA. And we now have two pieces instead of one. And now we just do it all again. Zip the two of them apart, set it to 55 degrees so the primers find the beginning and the end. Set it to 72 degrees and let the duplication do its work. Have all those individual letters snap into place. And now we have 4 copies. And it just keeps going and going and splitting again and again. And if you do the math, if you do it 30 times, 2 to the power of 30 is over 1 billion. And that's how you end up with over 1 billion pieces of DNA only cycling 30 times. And it only takes about 45 minutes. And now here's where things get crazy and copy over and over and over again. And you'll see how quickly the number of copies goes up with every cycle. A million of them, 4 million of them, 30 million, 100 million, 1 billion. And there it is. Let's go back and share the presentation again. There we go. Okay, I hope that played okay and didn't break up too much, and that was of some use to everybody. Okay, so now what? We have 1 billion copies of the exact part of the DNA that we wanted, but they're still too small to look at. These are tiny molecules. You can't look through a microscope and see the individual electrons that make up these things. So what do we do next? We do the process again with just a couple of special nucleotides sprinkled in. We make a special version of A, which we call A prime. And what that does is, if it ever gets used to duplicate a piece of DNA, if instead of the regular A, our special A prime gets used, it stops. It's missing the link on its right-hand side that lets other things attach to it. So it will stop the duplication early before it's done, and no more characters can connect after it. And you do the same thing with C, G, and T. So when you duplicate your DNA this way, you will randomly get pieces of your DNA of every possible size. If your DNA starts with C and the very first letter that got used was one of your special C's that doesn't let anything else get copied after it, you'll have only one tiny little C floating around and nothing after it. So at the end of this process, you have every possible length of DNA from 1 all the way up to 700 or so, and how long the area of DNA that you're interested in. And the last interesting thing that you do is you put a little drop of dye in them so that they kind of glow in the dark. The A's will glow yellow, the C's will glow blue, the G green, and the T red. Just a little bit. It's not enough to really see with the naked eye one at a time, but there's just a little bit of dye in there. So then you get this big block of jello, which we call an electrified gel. And you put all of the strands of DNA that you made at the left-hand side, and you charge that negatively, and then you charge the other side of the jello positively. And all of these ions and strands of DNA have a negative charge, and they're all going to be attracted to the other side of the gel. So all of these strands of DNA will start swimming up at the screen through this gel towards the top. And the interesting thing that makes this all work is that lighter things can swim faster because they don't weigh as much. So for instance, the tiny piece of DNA that's only the letter C is going to be the fastest. It's going to be able to make it all the way to the end of all the way to the top of the gel. The second, we only have 2 molecules with it, is the second lightest, and it almost makes it all the way to the end, but not quite. And then finally, the really big ones that were heavy are not going to be able to travel nearly as far. So when you have 1 billion pieces of this C DNA with a special dye in it, all in the same place, it's going to be enough to actually be able to see that color, and it actually makes a band in the gel. So this is an actual photograph of what your chunk of jello will look like after you turn the electricity on for a while. You'll have all these bands, and you'll see that this traveled the furthest. This is the band that made it all the way to the top of the gel, which means that it was the lightest, which means that it was only 1 character long. And since it's where you dumped all of the C's, you know that it's a C. So the very first character of our DNA is C. And the second fastest one was in the A lane. So the second character is an A. And you can look at this gel and you can read the DNA off of it. It's C, A, G, T, G, A, C, and you can see what it is. And you don't even have to read it by hand. You can take a picture of it and the computer can just look at it and tell you what it is. So I realized that this was kind of a long technical explanation about what happens, and I hope that it was of some value to everybody. I don't expect anybody to, I don't expect everybody to have caught all of the details of what was going on there, but I do hope that this has at least given some insight into how this works. And so that that it has been of interest to sort of see how this process works and how it's actually done in the lab. So another thing you can do is if they're dyed different colors, you can actually see all the different colors of DNA and just look at their colors to see what they are. So you would read this DNA as C, A, G, T, et cetera, et cetera. And as I said, the computer can just read it for you automatically. Now, sometimes this happens. You'll see there's 2 black bars at the very top, both C and G. What does that mean? It means that the very first character of your DNA is both a C and a G. Because remember, I was saying that there's actually 2 copies of DNA inside every person and every mushroom, describing 2 different organisms. And every once in a while, 1 copy is different than the other one. And if that happens, you will actually see 2 of the lanes at the same time, showing you that one of the copies of DNA was different than the other copy the DNA. And so you can make a statistical algorithm and take all these DNA sequences and make a tree about how they're actually related to each other. You can figure out the percent that they're related to each other, but how they evolved and the exact order of the tree is still just a best guess. It's not a magic bullet that tells you for sure how everything evolved from everything else. It still sort of just gives you a statistical best guess of how things work. So how much does this cost? Well, the first time they did it, the first time they sequenced DNA, it was probably like a $10 million project. And nowadays you can send your piece of mushroom away, and for somewhere between $20 and $40, you get your sequencing. So it's really come down in price. This is pretty amazing that it's accessible to so easily now, which is what's making this so exciting, that everybody can do it. Clubs can sequence their own mushrooms, and we can learn so much about this. And you can almost do it yourself. Step one, the PCR that the video was about, where you make 1 billion copies of the piece of DNA you're interested in. All the machine and the chemicals and the tubes, you might be able to get something like that for around $1,000, which is pretty amazing. Now, the second part that uses the gel machines and the electrolysis, that's kind of a little more complicated. That costs a few $1,000s, and it's not something that is so easy quite to do at home yet. It's still something that people that do step one in their house are still going to usually send out for a lab to do step two. But I predict that it will not be long before you can do all of these things in your home very easily and cheaply. So let's talk now, put all sort of the technical science aside and take a look at some of the interesting results that we've got from this. What have we found out? Let's play the match game. Here are 8 mushrooms. And the goal of this game is to figure out which pairs are actually related to each other, because there's really 4 pairs of closely related mushrooms here. And what you're probably going to guess, and you wouldn't be alone, is, well, first of all, this Russula are both guild mushrooms and nothing else is. So you might think that those 2 are a pair of closely related mushrooms. Secondly, these 2 kind of look like coral, this coral fungus and this really weird coralized puffball. So you would be forgiven if you thought that those 2 were related to each other. Chanterelles, the real chanterelle and one of the false hinges underneath the cap instead of gills. And they both have that. And it used to be thought that these 2 were related. And finally, the teeth mushroom, where you look underneath the cap and you see the spines. This is just the lion's mane, the bundle of spines just growing on a tree, and these are more cap and stemmed mushrooms that have these little spines and teeth growing underneath their cap. Well, you would not be alone if that's how you thought they should classify them. Since the 1700s, everybody has assumed that this is what the truth was, and it's really only in the last 20 years or so that DNA studies have shown us the astonishing truth, which is what they look like might have nothing to do with how related they are. The bear's head is order and are somewhat closely related to each other. The second pair is the agaricus and this puffball. Puffballs are fairly closely related to the portobello type of agaricus mushrooms that you find in the stores. And coral mushrooms and the woolly false chanterelle are actually also very closely related. They're even in the same family. And lastly, have you ever thought you found a chanterelle and you go and pick it and turn it upside down and it's really a hedgehog? They really do look a lot the same. And it turns out they really are fairly closely related too and taste just as good. So the hedgehog and the chanterelle are our last pair of closely related mushrooms. So now remember, for some of these, we did have a clue. The microscope did give us a hint about how this worked. Mushrooms have evolved back and forth between having gills, pores, and spines all the time. whether or not you're a gilled mushroom or a pored mushroom is not all that meaningful. And if you think about it, those differences are not as big as it might seem. If you were to take a gilled mushroom and every inch along the gills, you pinch them together again, you can kind of make pores out of gills. If you have a porcini and you tear into those pores, and you'll end up getting all these sort of tattered flesh hanging down that kind of looks like a spine, and you've turned it into a spine mushroom. Because it turns out that there's really only so many shapes that a mushroom can have and be successful in reproducing and be successful in making millions and billions of spores all over the fruiting body to be able to get those spores out there into the world. Gills work very well for that. Pores work very well. Teeth work very well. So as these mushrooms evolve, they have to find one of these really good successful shapes. So what do you do now? Great, you got your DNA sequence. So you just Google it on the internet, it'll tell you what the mushroom is, right? Well, unfortunately, no. The hardest part of this entire process is interpreting the results. You could write a whole course worth of material about how to take the DNA sample and figure out what you have. The problem is, we do have places on the internet like the Gene Bank and Unite where everybody stores their DNA results and you can compare yours to their DNA. And we also have this cool little web program called BLAST that will compare your DNA to the giant internet library of everybody else's DNA, or you can give it a specific set of sequences and tell it to make me a tree out of these, and it will do that. But the problem is, most of the mushrooms with sequences on the internet are misidentified. So if you see that your DNA sequence is the same as Russula zarampolina in Finland, that is probably misidentified, and it does not mean that your mushroom is Russula zarampolina. The only way to know for sure is for every mushroom species that there is, we have created and declared an official type specimen, the sort of official black morel, the one real one. And if you sequence the type, the real one, then you know for sure that it's that mushroom. And if you match that sequence, that's what you have. But we have not yet been able to take mushrooms because a lot of them are 100 years old or older, and you just can't sequence them. So it creates a lot of problems. And more problems is we might have sequenced the ITS region, which is a very short piece of the 50 million. Somebody else might have sequenced a different part of the DNA, and you can't compare yours to theirs unless you both sequence the same part of the DNA. And a lot of people ask, okay, if your DNA matches 100%, it's probably the same thing, but how different does it have to be before you consider that your mushroom is going to be different? This is still a matter of opinion. You have to look at all these differences by hand, and it just depends. You have to look at all of the physical, the microscopic, and the ecological information you have and put it all together. It's very important that I want everyone to understand that DNA is not this magic bullet that with a sequence you can figure it out. It is just one tool of many. And all of the other old tools of writing the descriptions and looking under the microscope and seeing the trees that are nearby are every bit as important as they always were. And DNA is now just one more piece of information that we have to help us get the whole picture. Now, there's a couple of organizations that are really doing a lot to help local clubs be able to figure out what mushrooms that they have in there. It was formerly called Mycoflora, is a combination of the amateur mushroom society NAMA and the professionals at MSA, and they have teamed up to create this this nonprofit called Fungus, which can do some great things. Their goal is to have a map of all the North American fungi, what they are and where they grow, and they can give guidance and instructions to every club on how to do DNA projects so that all 80 clubs across the country don't have to reinvent the wheel. sort of figure out some good practices, and they can help each club along. They provide some grant money so you can afford to do it, and they're trying real hard to find as cheap sequencing services as they can, but there aren't very many of those, and that's proving to be difficult. And they will also help make sure that your citizen science results get in the hands of the right experts so that you can really contribute in a major way to some papers being written and to our understanding by getting your information to the right people. But the biggest missing piece so far is that there are few people trained to study DNA and interpret what they see. There's no textbook yet. I was speaking to some grad students who said they kind of just talk to each other in the lab and they say, hey, I think you do this, let me show you. Not even a textbook on it yet. It's kind of like learning about the birds and the bees on the street. You kind of have to go in the lab and teach each other. So this is a very, very new technology. Now, MycoMap is another great website that coordinates with them to provide a single place for you to put your DNA sequences, your photos, the species distribution of where they all grow, so that you have one place to go and look and find all the information about a mushroom instead of having them scattered all over the place in so many different areas. And iNaturalist, as many of you know, is a really fabulous website. It has a cell phone app, so you can just take a picture on your cell phone, press a button and upload it, and then a whole bunch of other mushroom geeks will help you identify what it is. And that information will now be available for everyone for their studies. Let's talk lastly now just about a couple of some of the interesting results that have come out recently. Now, the deer mushroom. This is a pink spored mushroom. Pink spores are a really great spore color to find when you're looking at a guild mushroom, because there aren't a heck of a lot of pink spored mushrooms. There's millions of brown spored mushrooms and millions of white spored mushrooms. And when you find that, it can still be kind of daunting to try and figure out what mushroom you have. But you find pink spores and you really have a shot, because there are two families really of pink spored mushrooms. The Pluteus family, which the deer mushroom is in, have free gills that don't actually attach to the stem. There's a little space here, and they grow attached to wood on this moss-covered log here. And the other family, the Antoloma family, is the opposite. The gills do attach to the stem, and they usually grow on the ground. So it can be pretty quick to get which family you have of spored mushrooms. So there's a recent study that found that there's not only the deer mushroom, Pluteus urbanus, but there's a cryptic second species, Pluteus exilis, described from California, and they're super hard to tell apart. You almost actually need to do a DNA sequence to figure it out. They're very, very similar. So they did this study where they sequenced all of the deer mushrooms they could up and down the coast. And that California had both species. There were lots of sequences, but they only had 4 from north of California in Oregon and Washington, and all 4 of the sequences from the Pacific Northwest were Pluteus exilis, and they could not find the actual deer mushroom, Pluteus urbanus. So they concluded tentatively that it looks like up north you have Pluteus exilis, and in California you have both. So I wanted to test that theory. So I went around and found all the deer mushrooms I could, and the first 5 Pluteus that I found in the Pacific Northwest, in Salem and around Seattle and from various places, all 5 of them turned out to be Pluteus urbanus, and I could not find Pluteus exilis. So what does that mean? It means depending on who you listen to, the paper or me, it was a different species that was the only West. The paper implied you're only going to see exilis in the Northwest. My own little study said you're only going to find urbanus. Since then, I have found both. But what's the lesson here? The lesson here is studies with small sample sizes are dangerous, and you have to be careful what kind of conclusions you draw from them, because sequencing just 4 or 5 mushrooms is not enough to actually tell you what's going on. Another thing we found out is about Amanita muscaria. So the real Amanita muscaria is this pure mushroom with whiteboard that grows in Europe. And you do occasionally see it under introduced trees in North America along the West Coast here, but mostly not. What we call, and what you guys call Amanita muscaria subspecies flavivulvata, the one with these sort of a yellowish tinge to their warts, is the species that grows here. And its sequence is quite genetically different from muscaria. It's not a subspecies, it's not a sister species. It needs its own name. And we found its own name. It turns out that this beautiful Amanita in so many colors. It comes in, as you see here, red, orange, yellow, and white, have the same DNA. They're exactly the same. It does not matter what color it is. It must be some kind of environmental thing because it's not genetic. The exact same DNA sequence could imply any of these colors. So around about 100 years ago, somebody in Eastern North America described the white Amanita muscaria as Amanita chrysoblema. And it turns out that that's the DNA that matches all of our muscarias along the West Coast with our native trees, with the exception of a few recorded instances around some introduced trees of the actual Amanita muscaria. Our Amanita is a different species, and we can't really call it subspecies flavivulvata because it's quite distantly related and does not it's a genuine species in its own right. So if you want to be technically correct, thanks to new DNA discoveries, we should be calling our Amanita chrysoblema. That is the only species level name available for what it is. Now, should we do that? Maybe not. Somebody should maybe come along and publish some kind of name or make a paper to sort this out and find something that we can all agree with. But you would not be incorrect to Amanita chrysoblema because it's not Amanita muscaria. Another interesting thing, one example is we have sequenced Copernellus micaceus, the mica inky cap, all over the world, and the DNA is the same all over the world. And this kind of makes sense because urban saprobic mushrooms that just basically eat any kind of dead vegetable matter you give it, they're not very fussy, they tend to spread around the world quickly. So you can get some spores on your shoes, bring it home, and it'll start growing in your garden when you walk in your garden. It's very easy to spread because it doesn't really care very much what is that the mushrooms that are not mycorrhizal, mushrooms that are do not have that symbiotic relationship with the trees, are able to travel freely all over the world, and often it'll be the same species worldwide. But as I just explained with the Amanita muscaria, this is a mycorrhizal mushroom that lives in symbiosis with very particular trees. Well, we don't have the same trees here as we do in Europe. So bringing that Amanita muscaria from Europe here, even by accident, it's not necessarily going to find a tree it likes, and it's not going to really find a home here. So the mycorrhizal mushrooms often end up being different in the West Coast and the East Coast. And in mushrooms that are simply saprobic, we will find that they pretty easily spread all over the world. Now, there's of course a lot of counterexamples to each of these, but it does make for a really interesting realization that ecology is turning out to be one of the most important things of all when correctly classifying mushrooms. And I have a very interesting story to sort of really send that idea home. In the beginning, 100 years ago, when they were classifying the white-spored mushrooms, they came up with, among other things, two different genera, Trichiloma and Armillaria. They were both pretty beefy and A lot of them were two important differences between the two of them. And it's always great when you have two differences, because if there's only one difference, you don't really feel like that's a good way to separate them. But if you can always see that there's always at least two differences between them, you really feel confident that you've got two different things you're talking about. So they had tricholoma on the right. These do not have a partial veil. There's never that a ring around the stem, that piece of flesh that connects the outside of the stem. And they grow in the ground, coming out of the soil. And on the other hand, they described armillaria, and they do have that veil. You can see the ring left on the stem here, and they grow sticking out of a piece of wood right on the trunk of the wood. And with two consistent differences, they thought they pretty much had this covered. They knew what a tricholoma was and what an armillaria was. And then along comes the matsutake. What the heck is a matsutake? It grows on the ground like a tricholoma, but it has a veil like an armillaria. So they had to decide which of the two was it. And they had to decide if whether or not it was growing, touching a piece of wood or not, was more important than whether or not it had evolved this extra limb, this veil. And everybody thought, well, it can't be that important whether it is touching a piece of wood when it's growing or not. Like, why would that matter? But look at this. It has this extra part, this extra body part that other mushrooms don't have, this veil that grows between the edge of the cap and the stem. Like, that's a much bigger deal. So they decided that it was an armillaria, and the very first classification of the matsutake was in the genus Armillaria. Well, as you probably realize now, it is a tricholoma. They did the DNA, and they did other studies earlier, and they realized that it actually belongs in tricholoma. And the reason is that the ecology is the most important thing. It is a big deal if you're growing on the soil or if you're touching a piece of wood, because it implies an entirely different way of life. These matsutakes are mycorrhizal. They have evolved this symbiotic relationship with the tree roots, where they grow around the tree roots and they exchange sugar and nutrients in this whole system it's incredibly complicated. And the armillaria that's growing in a piece of wood, it's just growing fibers in a piece of wood and eating dead wood, and it doesn't have any of that kind of system. So the ecology turns out, once again, to be one of the most important things that you can know if you really want to properly classify mushrooms. It all comes back to ecology. Was it all worth it? Scientists, starting with Fries right up until the present day, some of them have spent their entire lives dedicated to drawing a tree of life to show how these mushrooms are related. They dedicated their lives to this, and they never thought they would ever get an answer. Here's a quote from Hessler and Smith's Waxy Cat book, the Hygrophorus book from 1963, where they were arguing about whether or not a mushroom belonged in this branch or in this branch. Was it more closely related to this one thing or the other? And they said, this matter will probably continue to remain one of opinion as it has in the past. And elsewhere they said, the end is just not yet in sight. Well, now, thanks to DNA, the end is in sight. I mean, it's still a matter of opinion how much difference of DNA means, and you have to look at the ecology especially and under the microscope and to sort of synthesize all this data. But pretty soon we really will finally know which mushrooms are actually related to the other ones. It's really fascinating to look back at these old books where they made decisions like placing the matsutake in armillaria, seeing where they were right and where they were wrong, and seeing how if they didn't follow the ecology, they were really wrong. And having dedicated their lives to this thing, I just sometimes wish I could go back and tell them that we know the answer now, because I'm sure they would love to live in this age where for just a few dollars, anyone can DNA sequence something and get not the whole answer, but that kind of last missing piece that with everything else, can be synthesized into finally answering some of the questions that have been plaguing us for hundreds of years. So thank you very much. I appreciate everyone who sat through that, and I'm now here to take any questions that you might have.
Okay, and I just told Phil they can unmute if they'd like to ask a question. They can
Absolutely.
ask it in the chat. Some comments from the YouTube group. There's a YouTube group. They found very interesting, particularly the part about the different colors of the Amanita muscaria or not muscaria.
Yeah, so what do we call the one behind you?
That's a good question.
Every time I look, it looks like it's getting a little closer. I'd be keeping an eye out over your shoulder if I were you.
Yeah, or I'm getting further. Yeah, we'll have to ask the artist what that was patterned on. And one question I had is, do we have anything here truly? Well, you said with the introduced species, we have some true Amanita muscaria. Because I know in Santa Cruz, there are several areas that, every year produce some very deep, dark red Amanita muscaria. But I guess the thing to look for is the color of the warts. Is that correct? Or?
Yeah, it's not foolproof like everything, but more often than not, a pure white wart tends to indicate the true European species, and ours has that sort of yellowish tinge to the warts.
Yeah.
Except in the pure white version, something gets rid of all the pigment, and it's pure white, and that includes the pigment on the warts.
On the warts. Okay.
So our natural white species has pure white warts because all of the pigment for whatever region has been bleached out.
Seen the white ones on the West Coast, Danny?
Pardon me?
Have you seen the white ones on the West Coast?
Yeah, we it's sort of the least common color, but I usually see one every year.
Okay, I've only seen them back east.
Okay, and Dora had a question about, I don't know if she wants to ask herself, does rot alter the DNA sequences?
Good question. Well, when it's rotted, unfortunately, it's usually other mushrooms like molds, which are mushrooms that are rotting your mushroom. So it doesn't actually change the DNA of the mushroom you're interested in, but it puts its own DNA everywhere. And what you accidentally end up doing is sequencing that mold, and you get a DNA sequence of mold. Now, most molds are ascomycetes, like morels. So if you're wanting to sequence a basidiomycete, you can actually do a more specific primer that doesn't just find mushrooms, but finds basidios in particular, and that will ignore molds. So you will easily be able to, more easily be able to get a DNA sequence out of a regular guild mushroom, no matter if it's covered with mold. Now, the problem is if your morel is covered with mold and you want to sequence that, they're both ascomycetes. So it's going to be a lot harder to separate your mold from your morel.
Okay, we have a YouTube question. I was going to put it into the chat, but let me just read it to you. Ann Goddard asks, is the Northwest Key Council keeping track of the sequencing and names along with the keys as they continue to be updated? And is this going online?
The Key Council is just getting into DNA now. We've started just before the pandemic, started doing a few DNA sequences, so that's sort of relatively new. So we will be, yes. Right now, I'm kind of doing it personally. I personally have a website where all of the DNA that I can see anyone doing from across the Pacific Northwest is being uploaded. And I can type that into the chat here. Let me put the website here. It's soon going to be part of PSMS's website, psms.org. But right now, it is a personal website, and there it is. It's a work in progress, so you can click on your favorite genus, and only less than a third or a quarter of them are clickable. I just don't have the data yet for most of them. But then you'll get to see a sort of a complete list of what species we have by DNA and what DNA has taught us in the last few years about that genus.
Okay. Yeah, I'll pass this on to the YouTube group.
Somebody asked about the lobster mushroom. Yes, because the lobster is an ascomycete infecting a russula. And I have seen people be able to both get the sequence of the lobster and the sequence of the russula out of it. So it does interfere, but if you know what you're doing and it's a relatively fresh specimen, then you should be able to and find the unadulterated piece of russula DNA and still be able to figure out what it is. Another thing that's interesting is people often ask, what are the odds of the lobster infecting a poisonous russula? Because there's no way you can tell what russula that used to be. It's all orange and pimply now, right? There's like, I don't care how good your ID skills are. You're not going to look at the lobster and say, oh, I see what that used to be. And no one's ever been poisoned by one. Well, the Russula brevipies group is a group of maybe a dozen species, and it's quite distantly related from all the other ones. Even Russula cascadensis up in Oregon, the sort of small, really hot, awful tasting one that looks almost the same, is pretty distantly related. So it's quite plausible that the lobster mushroom is only infecting the sort of not too spicy, not at all poisonous russulas, because they are isolated by DNA enough that I can imagine that there's no danger of the lobster infecting anything that you shouldn't be eating.
Well, clearly no one has ever worried about it because they sell them all over the place.
Right. But now it's okay not to worry about it.
Yeah. Oh, Danny, you'll love this question from David Kurtz. Is there a MycoMatch iOS app for California?
Yes. Well, one at a time. We're finally getting cell phone versions of MycoMatch. There is on micromatch.com, there's the beta version of an iPhone app. And Paul Hill in our club has been working very hard, volunteering time, making an Android version. And that's just about ready for people to try. So very, very soon now, probably before the next spring season, you'll be able to get an Android phone version, but it's still only Pacific Northwest. We will need clever volunteer people from California to actually put the data in for California, because we know we don't know enough to be able to do that. So we're going to need some hardy volunteers to help us with that. But one thing about MycoMatch is it is written in a way that anybody can, through the graphical interface, enter new mushrooms and set it up for another region. I'm not going to lie, it's a heck of a lot of work, but you don't have to be a programmer. You can go through the menus and you can put all your California mushrooms in and make a version for a different region. So it's certainly possible. And if you're interested in that kind of thing, certainly contact me. I see a question about what technology I for now. So I just gave one example of how to sequence. Once you've replicated the DNA, so you have 1 billion copies of it, you don't have to use the Jello method. There's other ways that you can do it. There's this thing called an Oxford nanopore, which is this machine that has these tiny microscopic holes that only small pieces of DNA can fit through. And as the DNA goes through the hole, it can figure out how long it is and read it for you. And it's something like only 60% accurate. But if you do it 1 billion times, you'll eventually see what sequence you got more often than you got any other sequence, and you can find out what, and then they wear out. You can like use this machine for a few weeks, then you have to like throw away the filter and buy a new one. So it's this completely different way of DNA sequencing. It's amazing the technologies people are coming up with. This is really what I talked about, is sort of the first and the most popular, but it's by all means not the only way you can do it. And people are coming out with new ways all the time. I'm personally, I'm sure we'll have a tray. I want to push a button. I want the tray to come out. I want to put it in the tray, close it. I want it to go beep, 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 and say Amanita muscaria. So that's what we're all waiting for.
Okay.
And the field version.
Right.
Yeah, something that attaches to your phone.
Right.
Let's see, I just dumped a collection of questions from the YouTube group. Jeffrey Souza, any new information on the genus Hypomyces and how it reproduces?
I do not know.
Okay, and Christopher Fleming, is it correct that you can run DNA amplification for varying sizes of base pairs? If so, does a larger set of base pairs help narrow down the true ID?
Yes.
Yeah.
So what
Matter of
you do
money
is the ITS region is great for telling if two mushrooms are the same species or a different species, but it's not so good for other things. We found sequences of mushrooms that we didn't know what it was, and the sequence is nowhere near anything on GenBank. No one else has ever sequenced anything like it before. We're like, holy cow, we found a new genus. We might have even found a new family. So what do? Well, you do sequence a different gene, one that mutates slower, and a different gene is useful for helping you find the genus and the family it fits into. And best of all, if you sequence 4 or 6 different genes and you combine them all together, the longer you sequence, the better results you get. So the studies that are trying to sort out the families are having to actually sequence 4 or 6 different genes. There's some very, very long pieces to be able to put that information together, because it's true. What sort of sequencing just the ITS piece is very good if somebody else has sequenced it, and you can see if yours is 100% match or a 99% match, and you can figure out if it's the same or not. But for the deeper questions of, is this a new genus? What family is it in? You have to sequence more than that.
Kind of a follow-up on that, Mr. Miller, are the ITS sequences in the mushrooms kind of similar to introns in the human genome?
I don't know. I don't know much about, I've heard of it, but I don't really know much about that to be able to say. Like, you have an example of what?
Just kind of areas of the human genome that like can be messed up and they're not going to actually alter, you know.
Right.
Right.
Okay. Right. It's kind of like a junk piece. So the fact that it is free to mutate without hurting the organism, it mutates faster than other things. Gotcha. And that's why it's the gene that's best of seeing if two things are the same or not, because you'll see every little difference between them. But there's so many differences between mushrooms in different families. So it's all completely different, and it's just too different to be able to sort out. So you need to also sequence genes that don't change very much, so that one change is very meaningful, and there'll be fewer of those kinds of changes. So you'll be able to get your head around what's going on, comparing things in different genera.
I see. Thank you.
I have a question. I think 2 months ago, we had a talk from Alan Rockefeller, and he was talking about psilocybe, and there was actually a question posed to him that was saying, you know, a lot of these species in this genus were historically placed in various other families, and how is it that all of these mushrooms were classified so strangely historically when they all share very close sequences? And Alan's response was effectively that it's likely that the psilocybin gene complex was transferred throughout all these mushrooms, potentially asexually. Do you have any?
You dropped out there.
Oh, sorry, I got muted there for a second.
Okay, yeah.
Just curious if you have any information about the asexual transmission of genetic information between mushrooms, or what the proposed mechanism of that change might be.
Sometimes you do see some very strange results, and we haven't really sorted out what it all means, or at least I haven't. So as far as the gene exchanges, you can find the same thing existing in a very different mushroom, but that doesn't usually affect the ITS too much, a very, very small percentage of the time. But what you do see is hybridization. You'll see two species will happen to be able to mate, and then their offspring, if you happen to find it and sequence it, it has the two strands of DNA that are normally the same are very, very different, like one from one species and one from the other. And it's a good question of how meaningful that is, because if it was enough of a mutant that it can never reproduce, it was just going to die out, then it doesn't matter. But if it was then able to go on and reproduce and make more children just like it, you get some pretty weird things in the gene pool that you find every once in a while. So we do see things like that happening when, and I don't understand enough about it to know what exactly is going on in all these cases. But there certainly are some strange things like that going on.
Very interesting. Thank you very much.
Okay. Oh, I had a question. Let's see if there's anything else in the chats. I don't see any.
Chat. I think we're caught up with the
I
chat.
think we're
which
all
is
caught
good.
up. Yeah. So, a lot of work with the human genome has, been, okay, trying to figure out what does this gene do, what does that gene do, and so forth. How far along are we with the fungus in figuring out what specific genes do?
Oh, very far from being done. Mushrooms are not getting a lot of study right now. Most people are focusing on trying to get them all sequenced so that we can know what they are. That's still, I think, a long way away. It's a very hard puzzle. It's kind of like, hacking into a bank and trying to reverse engineer somebody's password in it's kind of that kind of thing to figure out what this DNA means. It's very, very hard. Find all the hackers of the world and put them on the problem. That'd be the right kind of person to figure this out. But we're a very long way away from understanding that kind of thing.
Okay. Well, any more questions? I guess I should ask.
I hope that this was the right level of, showing some of this sort of scientific and talking about the repercussions and what it all means. I know there was a lot of technical stuff in the middle there, but I just sort of wanted to touch on all of the aspects of it so people can see what they're most interested in and what they might want to learn more about and sort of understand a bit about the whole picture.
Very helpful, Danny.
Good.
I really enjoyed it. Thank you.
So I guess one final item is that we should announce tomorrow night's speaker. The speaker I don't know, Maria Morrow, will be talking about the adventures of a microcosmonaut. She apparently is fixated on small things. So that should be an interesting talk.
Well, I mean, with everything that we've learned from the microscope and DNA, the smaller the apparent difference, the more important it is. Like, that's where all the really cool stuff is happening.
Yep. Okay, so that's same place, same time.
All right. Nice job, Danny.
Thank you. I
Yeah.
really appreciate it. Thanks for having me.
Yeah,
Yeah.
you bet. Good to see you again.
And this is the best commute I've ever had to have to come down and talk to you guys, I gotta say.
Well, hopefully, hopefully next year the fair will be an in-person event.
Be
again.
That would be great.
Yeah, and you didn't even have to shovel out your 20 feet of driveway.
Right.
You just let it sit
Yeah,
for now.
Yeah.
Thank you, everybody. I
You
really
bet. Good
appreciate
night.
it. This is really
Okay.
fun.
Yep. Good night. Thank you.
Okay.
Thank you, Danny.
Take care. Good night.
Good, everyone.